The overall goal of this procedure is to derive epithelial cells from three-dimensional mixed cell tissue cultures in order to analyze the effects of stromal fibroblasts on epithelial cell biology. This is accomplished by first establishing three-dimensional mixed cell spheroid co-cultures. The spheroids are then separated from the matrix culture in order to isolate the post-co-culture epithelial cells. The next step is to passage the post-co-culture epithelial cells until they are ready for analysis. Ultimately, the effects of exposure to fibroblasts in three-dimensional culture on epithelial cell behavior can be evaluated through microscopic imaging of the cultures and analysis of the post-co-culture epithelial cells, such as the fluorescence imaging of matrix invasion and transwell migration, as shown here.
Demonstrating the procedure will be Kun Xu, a research associate from my laboratory.
The day before establishing the co-cultures, remove the matrigel from the freezer and thaw it on ice in a 4°C refrigerator overnight. On the day of the experiment, place the culture medium on ice at least 1 hour prior to mixing it with the matrigel. To dilute the thawed matrigel, add half the volume of ice-cold HMAC medium necessary for the co-culture to a sterile tube on ice. Then, add the same half total volume of thawed matrigel to the tube to achieve a 1:1 dilution, keeping the tube on ice. Next, place 50 microliters of matrigel medium mixture mid-well in designated wells of a 24-well plate, and incubate the plate in a 37°C incubator with humidified air and 5% CO2 for